Hello students, good evening to all of y'all. Assalamu alaikum. Welcome to the online session. Can you all hear me clearly and see the screen clearly? Am I audible to you all?
Sir.
Yes, sir.
Okay, thank you. Yeah, I think the ones who didn't receive the link, I have put it in the group. You all can send it to your friends. Yeah, I think I can copy it from here. Yeah, I have sent the link. You all can share it to your friends because today is also a free session. So your friends also can join. Still some are sending the message. Now only some are registering. Give me a minute. Yeah, okay. Yeah, did you all receive the tute? Because as much as possible, we quickly send the tute.
No, sir.
Some of the students would have received it. The ones who
Yes,
received,
sir.
just raise the hand. Okay, yeah, still more to receive, no? I hope all of you all have filled that Google form. The ones who have not filled the Google form, later you all can go to the group, and in that there is a Google form. The last one which I sent, you all can fill it. If you all are not there in the group, no problem. Send me your name, grade, and school, and your area. My phone number is not 77828366. Then I can add you to the group, and from there you can fill the Google form. And after that, we will send it to you. Yeah, I'll send my number in the chats, no? Then it will be easy for you all. Thank you for. Save my number, and later on you all can send me a message. I will add you all to the group because first week was very hectic because new grade 10 admissions were there, new grade 9, new grade 11. So it was very hard to sort it out. So now only I have saved many of the students' numbers to my phone, and one by one we are trying to dispatch the tute. Most probably within next week we will try to complete all the tute sending things. By now about half have received the tutes, and the remaining ones, don't worry, you all will get it. Okay, don't worry, you all will get it. You all can contact me, and then after filling the Google form according to the address in the Google form, we will be sending it. Yeah, I have already uploaded the recording also of the first week. You all can watch the recording. In that we have given a nice introduction about, sorry, this is grade 11, no? I go for good. I've given a nice introduction about what is science, and like, you know, the importance of grade 10. All those things we have discussed in the opening day. Yes, I think we completed all the meristematic tissue, if I'm not wrong. Did we complete the meristematic tissues? Apical meristem, lateral meristem, intercalary meristem, all that we have done. Yes, now let's go into permanent tissues. If you all have the tutes, you all can check with the tute. Page number 5. Page number 5 in the tute. The recording, this free seminar recordings, it will be available for 2 weeks. Sometimes I will be telling them to keep it until the end of December. Sorry, end of January. So however, try to watch it. But normally once you purchase the classes, our normal classes, normal theory classes, for 1 whole week, the recording will be available. For 1 whole week, the recording will be available, and within that, anytime, any amount of time, you can watch the recordings. This is our grade 11 tute. I hope majority of you all got it. The others who didn't receive it, don't worry. We are still sorting it out. We became short with some of the tutes. I have printed it again, and we will be sending it to you all. Okay, don't worry, you will be getting it. Yes, now let's go into the permanent tissue. Now, what happens to this meristematic tissue? Can you all remember there were 3 types of meristematic tissue: apical meristem, lateral meristems, and intercalary meristems. Okay, apical meristem, lateral meristems, and intercalary meristems. So, what happens to these meristematic tissue with time? With time, the meristematic tissues grow, they become mature, and they differentiate into other types of tissues. So those are the permanent tissues. They become permanent tissues later on. And once they become permanent tissues, they have the ability to do a lot of functions. Once they grow into permanent tissue, they do not have the ability to divide further. That means growth is stopped. The growth is stopped, but they can perform a lot of specialized function. One by one, we will be discussing it today. I'll try my maximum to complete simple permanent tissues, and let's start a little bit related to the complex permanent tissue if we have time. Okay. There are 2 types of permanent tissue: simple permanent tissue and complex permanent tissue. In page number 5, in your tube, page number 5, you have 2 boxes. Permanent tissue. Under that, you have 2 boxes. One is simple permanent tissue. Yeah, it's already written. I'll put PT, simple permanent tissue. It's already written. One type of cells collected together, only one type of cells, no different types of cells, only one type. Who are they? PCS, that was our short form, no? Ali, ALI for meristematic tissue, PCS for simple permanent tissue. What are these PCS? Parenchyma, collenchyma, and sclerenchyma. Parenchyma tissue, collenchyma tissue, and sclerenchyma tissue. The next type of permanent tissue is complex permanent tissue. Under complex permanent tissue, it is made out of different types of cells. If you take the xylem, the example for complex permanent tissue are xylem and phloem. This xylem is made out of 4 different types of cells. All those 4 different types of cells together only forms the xylem. I told you last week also, I told you, just remember 4 letters: VTPF. Vessel elements, tracheids, parenchyma, fiber. V, T, P, and F. Vessel elements, tracheids, parenchyma, fiber. Those names you all will come across it later. We have to draw structures related to it. Don't worry. Just remember the first letters. VTPF, and phloem, it has 4 different types of cells. SCPF. So xylem VTPF, phloem SCPF, those 4. So page number 5, the basic namings are there. Then from page number 6, one by one we will be studying. Okay, one by one we will be discussing it. Hope all of you all are clear and settled. The ones who have the tutes, I hope your tutes are open and ready and you are looking at the screen, and whatever needed you all are writing. The others who don't have the tute, don't worry, you all will be receiving it as quick as possible. Because we were like, the full institute, because for the institute people, they are the ones who, I'm not the ones who is packing and sending the tute, the institute admin. For them, the maths tutes were there, new admissions for ICT, commerce, science, all the subjects, and physical class students also. This time, many came, more than expected. So it was a very rush week for the administration. So don't worry. Okay, we will sort it out, and within next week, we will try to send you all the tute. So until you get the tute, you all can just write certain things. Now, whatever I am writing from the pen, You all can write it down and keep, and once the tute comes, you all can fill it. I am putting with the page number. So once the tute comes, with the, you can check the page number and you can fill it. Hurry, shall we go into the discussion of parenchyma? One by one, we will start discussing parenchyma. Okay, when you look at parenchyma, most abundant type of the tissues in plants. That means out of apical meristem, lateral meristem, intercalary meristem, parenchyma, collenchyma, sclerenchyma, xylem and phloem, out of that 8, who's the highest? Parenchyma. This is in page number 6. Already the written things are already there in the print, so don't need to worry. Second point, they are saying the cells are isodiametric, spherical in shape. Intercellular spaces available. That means, I'll draw it in this corner, in this side. If you take a parenchyma tissue, it is made out of lot of cells. They are isodiametric in shape. Isodiametric means height and the length and the height are almost same. So it looks like a sphere, spherical. And they have intercellular spaces. The space between two cells is only known as intercellular space. Here we have drawn about 5 parenchyma cells. Collection of cells form the tissues, no? So when there are many parenchyma cells together, it is parenchyma tissue. Composed of living cells which form the soft part of the plant. Softest part is apical meristem, lateral meristem, intercalary meristem. Next to that, the soft part is parenchyma. There are large centralized vacuole. So this is a common feature of a plant cell, no? Normally a plant cell will have a large centralized vacuole. But in meristematic tissue, it was slightly different. Who was in the center in meristematic tissue? Can anyone remember? Who was in the center of meristematic tissue? Yes, in the chat box I have got some messages. Yes, a nucleus was there. But in parenchyma, which follows the normal plant cell category, it has a large centralized vacuole. And there is a small nucleus on the corner and thin cytoplasm. These are the cytoplasm, organelles in the cytoplasm. Nucleus and the other organelles. So we have a large centralized vacuole in the center, nucleus in the side, and all the other organelles and the cytoplasms in the corner. Thin primary cell wall with uniform thickness. The thickness of the cell wall is uniform throughout. Okay, it's same throughout. In some cells later you will study, one part is more thicker, other part is thinner, but here almost all are same thickness, uniform thickness. In page number 6, next to these parenchyma, these wordings, there is a small space. Can you all draw this? This is picture of parenchyma tissue. Collection of parenchyma cells form the parenchyma tissue. If you all have any doubts or any problems, you all can ask. Okay, done. Shall we go to the next part? You have a lot to draw today under parenchyma, collenchyma, sclerenchyma, if possible, xylem, phloem, have a lot to draw. Yes, the ones who didn't draw, take a screenshot. This is in page number 6. Then we have locations. That is very important. So the liquid around the vacuole is the cytoplasm. Yes, Pudha. That liquid around the cytoplasm is the around the vacuole is the cytoplasm. The cell wall doesn't differ from each other. No, it's same cell wall, uniform thickness. Okay, location balanna. Cortex and pith of dicot stem and monocot root. Here there are 4 locations. Cortex of dicot stem, cortex of monocot root. Pith of dicot stem, pith of monocot root. But easily you can remember in one wording, cortex and pith of dicot stem and monocot root. Later I will draw the dicot stem and show you all the parts, very important. Then second one, fleshy part of the fruit, and fourth one, endosperm of the seed. Those 2 locations we can draw it in one diagram. I'll draw the cortex and pith, don't worry. Okay, don't worry about this. Cortex and pith I will draw and show. First we'll draw the fleshy part of the fruit and endosperm of the seed. Listen to me and watch the drawing, okay? If you look at the seed of a fruit, okay, a fruit might have a seed. Inside the seed there are 2 parts. One part is known as the embryo part. Embryo. That part only grows into the plant. While the plant is growing, it can't do photosynthesis. When it is growing, no. When the seed is germinating, it can't do photosynthesis. So someone has to give the energy. Already stored energy is there. Stored part is there in the seed, that is known as endosperm. This endosperm is parenchyma. He will only give the energy to the embryo to later grow into the plant. Once the leaves come, they can produce their own food, own energy. Until the leaves come, this endosperm is helping the embryo. Endosperm is an example of parenchyma tissue. I hope you all are clear about endosperm. Then around the seed, we have the fleshy part. The flesh we can eat, no? That flesh is also made out of parenchyma. Fleshy part of fruit. So then the endosperm is the energy needed to grow. No, endosperm gives the energy. It is also a part. It's a white part like a seed part. It gives the energy. It gives the energy for the embryo to grow. So this fleshy part of the fruit is also parenchyma. So you can remember both these examples from one diagram. Page number 6. Next to the location, you have a space, no? If possible, draw this. Spelling of endosperm here, it is already given E-N-D-O, endosperm of seed. The writing part, already it is there in the tube. Only the drawing part, you need to draw it. I hope you all are clear about it, no? Okay, shall we go to the next examples? Can you all take a screenshot? Embryo is not parenchyma. Embryo is different tissue. Parenchyma is also known as endosperm. No, Buddha, listen to this carefully. Okay, very, very simple thing. Don't overtake it. Locations of parenchyma. Where is parenchyma? In the endosperm of the seed. In that, it's made out of parenchyma. Fleshy part of the fruit, it is made out of parenchyma. We'll study the other examples also. For now, we have discussed too deeply. Then, what are the other locations of parenchyma? Where can you find parenchyma? Leaf mesophyll, palisade and spongy. When I draw only, you will understand it clearly. If you take a leaf, here they have just shown you, fleshy part of the fruit. All this fleshy part has what tissue? Parenchyma tissue. It's here. I'll have to take some space and I'll draw it. Give me a minute. Yes, now I am going to explain this leaf mesophyll thing. So I want all of you all to look at the screen and follow this. If you take a leaf, okay, there is a upper part of the leaf and bottom part of the leaf. No, in between there are many structures. Even though it is a line, if you keep a microscope and see, there are many structures. What are those structures? If you look at the upper part of the leaf, we call it upper epidermis. Upper part of the leaf is upper epidermis. Lower part of the leaf is lower epidermis. So this is the upper epidermis. And this part is known as the lower epidermis. In between the upper epidermis and the lower epidermis, that palisade mesophyll and spongy mesophills are there. They have chloroplast, therefore they are green in color. This is known as palisade mesophyll. And then there are other some cells like this that is also parenchyma, made out of parenchyma tissues. Spongy mesophyll. Both these together only this is known as the leaf mesophyll. Are you clear? All these four names, if you know, that is enough, but I'll just give you one additional thing. Upper part of the leaf and bottom part of the leaf, when you see it, upper part is more shiny. Okay, just later go and check few leaves. Upper part is shiny, lower part is little dull. The upper part is shiny because there is a wax over the upper part of the leaf that is known as the waxy cuticle. Waxy cuticle. And also, another important thing is, in the lower part, lower epidermis, we have a lot of holes. Can you see these holes? What is this hole known as? That is the place where air is going into the leaf. Oxygen, carbon dioxide, Daytime carbon dioxide will go, oxygen will come out. Already you all have studied, even in grade 8 you all have studied. What is that? Stomata. Stomata. And guarding the stomatas, there are two cells. They only open and close the stomata. Now the stomata is open and later they can close. Open, close. Who guards the stomata? Guard cells. Guard cells. So all the details about the leaf is given. Waxy-cuticle, upper epidermis, palisade mesophyll. This is a type of parenchyma. Spongy mesophyll. It's a type of parenchyma. Then lower epidermis. In the lower epidermis, stomata is there. To guard the stomata, guard cells are there. Are you all clear? If you have any doubts, please ask. Yes. Quickly draw this. The ones who have the tute, you have page number 7. I have kept a one full space page. Just take about less than half, because we have another bigger diagram to draw. Less than half space and draw this. This is to show the location of parenchyma, palisade mesophyll, spongy mesophyll. I have given the full diagram. Upper epidermis, palisade mesophyll, spongy mesophyll, lower epidermis. Upper epidermis has a layer that is only waxy cuticle. Lower epidermis have the hole, those are the stomata. Guarding the stomata, we have guard cells. They can open and close the stomata. Once you're done, just send me a message. We can move to the next part. Okay, many have done. The others, you all can take a screenshot and you all can complete it later. Next we'll go to, now you all are okay with this leaf mesophyll thing, no? That is also made out of parenchyma, leaf mesophyll hari. The names are spongy mesophyll and palisade mesophyll. Now we'll go to the very important one, cortex and pith of dicot stem. Yeah. Last week also we drew the dicot stem. Today I will draw the whole thing and I will name it. So this is the dicot stem. We have to take a dicot plant like this, cut it this way and observe from top. Then we can see the rings. No, there is a plant. You cut it like this and observe from top. We will be able to see the rings. Okay. So the outermost layer is known as epidermis. In the leaf also we had the epidermis. Here also we have the epidermis. Inner to the epidermis, that area is known as cortex. In the cortex, we have triangular structures. You can remember like pizza. We have some triangular structures like this. These triangular structures are known as vascular bundle. In the middle of the vascular bundle, there is a ring like this. By now, you all know the name of that ring. What is that? What is the tissues present there?
Cambium.
Yeah, the cambium tissues. Okay. What type of tissue is cambium tissue? Lateral meristematic tissue. It falls under lateral meristems. The ones who are not there last week, you all can watch the recording, then you all will understand. This is falling under lateral meristematic tissue. Then the innermost part is known as pith. P-I-T-H. So in that example location, we said cortex and pith of dicot stem. So cortex is made out of parenchyma tissues. Pith is also made out of parenchyma tissue. Are you clear? Two locations of the parenchyma tissue. This is dicot stem. And also I'll just add one more point. The cambium is there now. This cambium divides the pizza into two parts. This part is towards the center xylem, towards the outside phloem. Xylem, phloem. Xylem and phloem. Xylem and phloem. Xylem, phloem. Xylem, phloem. In small grades you all have studied, xylem is transportation of water, phloem is translocation of food, no? So the barrier between both is the cambium ring. So epidermis, you all are okay with that? Cortex, okay. Vascular bundles, then the cambium, then the pith. Here our two main people are cortex and pith of dicot stem and monocot root. Here I have drawn the dicot stem. Monocot root you don't need to draw, but this is very important. Dicot stem is very important. Monocot root also, there is a cortex, there is a pith. There are also parenchyma is only there. This is related to dicot stem. Yes. Page number 7, you have the space. Please draw it. Draw a proper clear diagram. If you have any doubts, send me a chat box message. Yeah, there is a question, sir. Is the tute only for tuition students? No, Puta. The first 3 lessons I have put it into one tute. That tute is free for all. Any students who come, you all can get it. Send me a WhatsApp message. I will add you to the group. After filling the Google form, we will post it. It's a free tute from our side. For the first month, we are giving it free. The other months also it is free, but only for the class paid students. This time we are giving to all. Yes, any other doubts? Yeah. I'll just show the previous diagram. The ones who want to take a screenshot, quickly take it down. This is the palisade and spongy mesophyll example diagram. Take a screenshot of that. Now let's go to the dicot. Yeah, we have already gone. I'll quickly draw it. I hope you all are done with that, no? Yes. Any other doubts? Yeah, the ones who didn't take the ones who couldn't complete it, don't worry. Take a screenshot and you all can draw it later. This is the dicot stem. Here our main people we are discussing are the cortex and pith of the dicot stem. I hope you all are clear. Yes. Have you completed all the examples? Cortex and pith of dicot stem. Like that in the monocot root also it is there. You don't need to draw. This monocot root, you don't need to know the shape or anything. Dicot stem you should know. Just remember, monocot root also has cortex and pith. We are left with the last one, ground tissue of monocot stem. Dicot stem we drew. Now I am going to draw the monocot stem. Monocot stem is like coconut tree stem. If you cut the coconut tree and see from outside the vascular bundles, those vascular bundles are not arranged like the dicot one. It's just scattered. Here it's scattered vascular bundle. Okay, well now we'll name it. The outer covering as usual is known as epidermis. Inner to the epidermis we have ground tissue. Like earlier we had cortex, we have ground tissue and these are vascular bundles. Do these vascular bundles have a cambium part? No, cambium is only present in dicot, not in monocot. Last week we discussed that. Cambium is present only in dicot, not in monocot. This ground tissue has parenchyma, so it is made out of parenchyma. So this is the structure of the monocot stem. Monocot stem. Take this down also. Any doubts or any problems, please ask. Because actually, Putha, in grade 11, we are studying more deeply, little bit more deeper than grade 10. So you need the understanding. Okay. So if there is any doubts, please ask, because some students, they complain in school when they ask the doubt, they are getting scolded from the school teacher. Don't ask unwanted doubt, I told no. But here it's not like that. You ask a proper doubt, you ask the doubt, I will answer it. So is ground tissue present in dicot plants? No, no, Buddha. Instead of ground tissue, we had what in dicot plants? Cortex. Ground tissue and cortex are like same. In monocot, we call it ground tissue. In dicot, we call it cortex. Don't they have pith? No. In monocot, don't have that middle space. Here you can compare these two. There, because of this nice arrangement, there was a middle part, that is the pith. Outer corner parts are cortex. But here, the whole other parts are known as ground tissues. I'll just highlight it and show. These parts are the ground tissue, the whole thing. Except for the vascular bundle, all the other parts are ground tissue. Are you all clear about the ground tissue? Yeah, what is the function of vascular bundle? What does vascular bundle do? Yes. Actually, vascular bundles, okay, it's a bundle. Bundle of what? Xylem tissue and phloem tissue. Okay, if you take one vascular bundle, whether it is dicot or monocot, they have two parts. Xylem part and phloem part. What will the xylem part do? It transports water. Phloem part will transport food. So inside the vascular bundle, food transportation and water transportation is happening. Already in grade 10, you all studied, no? The sucrose is the food of the plant. Sucrose will be transported. It will be converted to glucose starch later, but when transporting, it is sucrose. Can you all remember? Disaccharide. Maltose, lactose, sucrose. Yeah. Once you all are done with this diagram, please let me know. Then we can go to our next issue. No, we have to go to the functions, no? Yes, one more doubt, Munada Balamu. Yeah. Yeah, this is the monocot stem. So see how clearly we have discussed the locations. Cortex and pith of dicot stem properly done. Monocot root I am not drawing. Just remember the names. Fleshy part of the fruit that we drew properly. Endosperm of the seed. Leaf mesophyll. The palisade spongy. And the ground tissue of monocot. Okay. Now see in this picture also you can see clearly, no? Epidermis, the upper epidermis, palisade mesophyll, then we have spongy mesophyll, and lower epidermis. In the lower epidermis, can you see the stomata? This is a 3D diagram. And the upper part is the waxy cuticle. It's when you go to the functions, what are the functions? Page number 6. I have already given the functions. Okay. When you look at the first function, photosynthesis, now you all know the place, exact place in which parenchyma is doing photosynthesis. Palisade mesophyll, spongy mesophyll in plant leaves contain chlorophyll within the chloroplast. Chloroplast is the organelle, chlorophyll is the green color pigment. What does that help to do? Photosynthesis. Therefore, photosynthesis takes place inside spongy mesophyll, palisade mesophyll. Therefore, we can say functions of some of the parenchyma is photosynthesis. Functions of some of the parenchyma is food storage. Food is stored in parenchyma tissue and they are called storage tissue. Example, potatoes. Potato tuber means the potato. Carrot, sweet potato roots. Pepper, banana fruit. All those, the food is stored in what? Parenchyma. So parenchyma acts as a food storage. It also can act as a water storage. Third function, it can act as a water storage. Especially this xerophytic plant. How do you pronounce this? Xerophytic. Xerophytic. Xerophytic plants, they store water. The parenchyma of that xerophytic plants, they can store water. What is these xerophytic plants? They are normally called desert plants. Desert plants. Aloe vera, bryophyllum, cactus. Those are examples of parenchyma tissues storing water. Parenchyma tissues storing water. Last one, provide support. Providing support. Here there is a new word, not a new word, already in grade 9 you all have studied. Herbaceous plant. Can you all remember we studied woody plants and non-woody plants? The non-woody plants are herbaceous plants. They absorb water, okay, they absorb water and become turgid. They absorb water and become turgid and they give the support of the plant. I will draw and show. Before going to that, another storage of parenchyma is air. So food, water and air is stored. Here they have not given the exact places. So just remember it can store food, water and air. Okay, it can store food, water and air. So if we write desert tissue instead of that name, desert plant, yes, you can write. Or you can give example, cactus. You all can remember cactus, no? Yeah, air storage, normally, there are some seeds of some water plants which are floating. For them to float, inside air should be there. So the parenchyma traps the air and then it floats. But in our O-level syllabus, they have not discussed much about it. Just remember, food storage, you know the examples. Water storage examples is there. Air storage, you can remember. Water plants, aquatic plants, they absorb the carbon dioxide from the water, they store and keep it. I hope you all are clear. This providing support part, I will explain it a little bit further. Balanna. If you don't water the non-woody plants, woody plants, they have a wood, they have a bark, they stay steady. But there are some plants, when you don't put the water, they are like dead, mehema. But once you put the water, they will become erect and they will, the leaves, everything will be proper. So we'll say this is the plant. Now it's like dead. Why? No water. But once you put the water, the plant becomes erect and the leaves spread out, and after adding water, same plant. So these are herbaceous plant. What happened? The water was absorbed into the parenchyma. Inside the parenchyma, we said vacuole is there. No? It got absorbed into the vacuole and the cells became blown up. Like balloons, it got blown up and it is now. So mechanical support is given by parenchyma. Now it's erect like this, no? Due to the mechanical support given by whom? Parenchyma tissues. How did they give the this one mechanical support? The parenchyma tissues, they absorb the water into the vacuole and vacuole got blown up and the cells became full, turgid. That is known as turgid. What if they say to draw the cross section of parenchyma? Can you draw and show? Already I have showed, no, first itself we drew the parenchyma tissue. I will again show it to you. Basic pattern of parenchyma tissue, we drew it. Any doubts about this turgidity? Okay. If there is anything, you all can ask. So you have to remember certain words. Herbaceous plants absorb water into the vacuoles of the parenchyma cell and become turgid. Therefore, it provides what? Mechanical support. Okay, mechanical support. Yes, the azan is going on. Once the azan stop, I will start the discussion. Give me 2 minutes. Yes, someone asked what is the structure of the parenchyma tissue. This is the structure of the parenchyma tissue. Here first itself we drew it. Yes, someone wants to know what is this turgid. Again, few students asked. Again, I'll tell what is turgid. Okay, normally if the vacuole is not filled with water, cell will be like this. When the vacuole is drinking water more and more, absorbing water, the vacuole will get blown up like this. Then it will be giving a support to the cell. This is only known as turgid or turgidity. It's like a balloon which is empty. When you fill water to the balloon, it will become big, no? High pressure, turgid. Clear about that? Now problems related to parenchyma done, no? We saw the structure of parenchyma, locations, we drew a lot of diagrams, and then the functions of parenchyma. Feature, function, location. All three you all should know. Good. Then collenchyma. Okay. First we'll read it one by one, and then next page I will draw all the needed things and show it to you. Cells are elongated, long, when you're seeing from side view. But when you're seeing from the top view, it is box shape, polygonal shape. Polygonal means something like this. When you see side view, it is elongated. I will nicely draw a 3D diagram and show, then you will understand it. Okay, that is known as cross section or transverse section. Page 7, what is there in page 7? Page 7 only you have to draw all the diagrams. That dicot stem, monocot root and all those things. Those diagrams. Now page number 8. In the TUT, if you have the TUT, page number 8. Cells are tightly packed, therefore no intercellular space. They are tightly packed, therefore no space in between the cells. They are living cells, large centralized vacuole, okay, thin peripheral cytoplasm with small nucleus, same like parenchyma. Because of the vacuole, the cytoplasm is pushed to the corner and a small nucleus is there. A common plant cell. Plant cells are like that, no? Primary cell wall is present. Okay, if possible, highlight this. But thickness is not uniform. Their thickness is not uniform. That is a specialty about this. Corners of the cell wall are more thickened by cellulose. Corners are more thickened. I will draw and I will show it to you. Corners are more thick. Okay. Now we'll go to the drawings. Are they going to ask the shape of parenchyma, collenchyma in exam? Yeah, once in the past paper, they have given the pictures and they have told to identify which one is parenchyma, collenchyma, sclerenchyma, and they have put some animal cells and confused us. Like that questions are there, possible is there. And also why we are drawing this is, all these details will easily be there in the diagram. So look at this, huh? The cells are polygonal in shape. The cells are polygonal and they are tightly packed. This is one cell, this is another cell, and this is another cell. They are polygonal in shape. This is when you see from top. When you see sideways, something like this. This is the top view, no? Sideways when you're seeing, it's elongated. Upper part is something like this. It's elongated like this. But when you cut like this and see, you will be able to see one of this. This is longitudinal section, LS, already given in the tube. When you see like this. TS is once you cut open and see it like this. For example, there is a person's head. When doing the surgery, they can cut like this. That is longitudinal. Or else they can cut like this and see the brain part. That is transverse. If they cut like this, it is transverse or cross section. If they cut like this, it is longitudinal. So from this diagram, we are able to understand that it is polygonal shape, tightly packed, no intercellular spaces. Then the cell wall. I'll just draw one cell wall and show it to you. Cell wall, some places it's like this. Some places it is more thick. Can you see? Here it's normal. Then now it's going to become more thicker. Can you see the thickness increasing? Then it is normal, small size. And then again it is increasing. Here again normal. And here more thickness. The corners are having more cellulose, more thickness. Again see this, huh? Thin, thin, thin. And now it's going to increase. Here increase. Thickness is more. So we cannot say that the cell wall is uniform. It's not uniform. In some places it is more thick. In some places it is thin. Some places it is thick. This is collenchyma. It's known as collenchyma. Does herbaceous plant mean herbs or all non-woody? Normally, all the non-woody plants which do not have a strong stem are falling under herbaceous category. Most of the herbs are herbaceous. So don't think herbaceous means only herbs. Most of the herbs which you are using as a medicinal plant are falling under herbaceous category. Not all. Okay. The ones who don't have a proper bark is only known as herbaceous. Okay, now here we have drawn the cell wall part. This is the cell wall part, okay? I just colored one. I can't color all those things. One is enough now for you all to understand. Now let's draw the vacuole. What about the vacuole? Large centralized vacuole. Nucleus is small. And we have a thin cytoplasm in the corner. Large centralized vacuole. Small nucleus. And all the organelles are in the side. Cytoplasm has all the other organelles, no? Can you all remember the organelles? Mitochondria, chloroplast, vacuole, Golgi, endoplasmic reticulum. We have rough endoplasmic reticulum, smooth endoplasmic reticulum, all the other organelles. Are you all clear about the collenchyma? Whatever we studied in the written format, now in the diagrammatic format, it is there. So when you remember the diagram, you will remember the features. So page number 8. Can you all quickly draw this? Page number 8. Next to the locations, next to the locations, there is a space. You can draw it. If you want, you can draw the LS. Mainly, we are worried about the TS, transverse section. TS is transverse section. I told you. For example, what can I show? If I take this mug and cut it like this and see from top, I can see the circle, no? That is transverse. But if I cut it like this, if I cut it like this, it will break into two parts. Those are the longitudinal section. Yes, quickly focus and draw this. If you all have space, yeah, you have space. Page number 8. The ones who have the tute, you can use page number 8. The others, you all can draw it. Once you get the tute, you all can draw it nicely in the tute. Did you all start the lessons in school, Lahan? Again, school holidays, no? Only for 1 week you all had school. Again, holidays. Again, you all will have school. Again, either Ramazan holidays or again O-level is starting, O-level holidays. So like that continuously, you all are getting holidays and schools, and after that they will say sport meet practice. Time will get wasted for that. International schools are there, that's good. Okay, then the locations of colon chyma, there are two locations. Forms a cylindrical ring inner to the epidermis in dicot stem. Can you all remember the previous diagram which you drew related to the dicot stem? Big diagram with the pizzas. I hope by now you all know the names, no? What is this name?
Epidermis.
Epidermis, very good. Inner to the epidermis, normally we have the cortex, no? Okay, cortex is there. This colon chyma is a small, in some plants, a small ring inner to the epidermis, there is colon chyma. Not much, a small ring. So that's why they have said inner to the epidermis of the dicot stem, who's there? Colon chyma. This is epidermis. Below that we have the cortex. In between the cortex and epidermis of some plants only, we have colon chyma. In young parts, the colon chyma is green color. That means it can do what? It can do photosynthesis. Give me 1 minute. Hello. I will clear about it. Inner to the epidermis of dicot stem. Inner to the epidermis. Next location is veins of the dicot leaf. So that means colon chyma is not present in monocot. I all clear. So location wise, you can draw two things. You can draw this part, this small line. Hello, Avila dinne. Hari brother, 5 minutes na mangenang. Okay, then the leaf veins. Leaf vein. They have colon chyma. Just draw these two structures. This one, you don't need to name all the parts. Just epidermis and you put cortex. In between the epidermis and cortex, who is there? Colon-chyma. Page number 8 itself, you have space. Above the functions, you all can draw it. Give me like 5 minutes. Your tutes have come. The printer has now only sent the tute. Okay, I'll go down and collect it and come. Yes, any questions? How can we get your tubes? How can we purchase? No, first tube, you don't need to purchase anything. Only thing is, send me a WhatsApp message with your name, grade, school, and the area. Then I will send you all a Google form once you added to the group. Then once you fill the Google form, it's easy for us to post it. Okay, now let's go to the functions of colon-chyma. Support and flexibility. Okay, colon-chyma, in the tute already I have mentioned this, listen to it carefully. Colon-chyma provides mechanical support to dicot stem, because colon-chyma is not there in monocot plants. Okay, therefore before the formation of wood, before the main wood parts come, this colon-chyma is only giving the support, even in herbaceous plants. And also if you take the leaf, I have given another point. The tissue provides support to the plant leaves by the colon-chyma in veins. If you take the vein, when you tear a leaf, it's easy to tear, but in this vein part, it's little bit hard. And especially in this mid-rib, mid-vein, it's very hard because there is a lot of colon-chyma. So that means colon-chyma is giving little bit of a strength, no? That is only known as support. And the leaves and the stem can bend. That is flexibility. That is also due to colon-chyma. Photosynthesis, second function. First function, support and flexibility in herbaceous plants. Second one, photosynthesis. Because there is chloroplast inside the colon-chyma, okay, if you take the stem, branches or the stem or whatever, in the initial stage, they are light green in color, no? The stems are initially light green. Later on, it becomes brown. Initially, it is green. That green is due to that colon-chyma ring. That colon-chyma ring gives the green color because it has chloroplast. Therefore, it can do photosynthesis. Later, it will become brown. At that time, it can't do the photosynthesis. But initially, when the plant is very young, even the stems are green, even the stems will help for photosynthesis. That is the colon-chyma tissue. So you can easily remember this chart. Wait. If you look at a plant, there are two main types of plants, no? Dicot plant, monocot plant. Is colon-chyma present in monocot plant? No. In dicot plant, we'll take the main three parts: leaf, stem, and root. Does the root have? No, we didn't study anything related to colon-chyma. That means colon-chyma is there in the leaf of dicot, stem of dicot. Leaf of dicot, stem of dicot. If I ask you the question, where in the leaf, you can easily say leaf vein. Leaf vein. Where in the stem? Inner to the epidermis. Inner to the epidermis. I hope you all are clear about it. Shall we move to sclerenchyma? Parenchyma done, colon-chyma done. Now we are moving to sclerenchyma. Yeah, someone is asking in the WhatsApp, will the recordings be provided? Yes, Puta. The free classes, I am giving all the recordings to all the students. Normally, once you purchase the classes, my class students, they will get a personalized recording in the website. And through that, you can watch the recordings within 1 week. You can watch. Before going to the next part, I think it's better for me to introduce the website. No, some students don't know about the website. Our website's name is masteronline.lk. Okay, masteronline.lk. Masteronline.lk. When you go to masteronline.lk, you will get a screen like this. Just for the new students, I am saying, because some of them, they don't know. First, you have to sign in, register. For that, they will ask your name, grade, school, address. Put all those proper address. Then only the tudes will be, the future tudes will be sent to that address. This is how to purchase the class. And then after you complete the registration, your proper email will be there, proper phone number will be there. They will send a code to that. And after verifying everything, then what will happen? Then you can go to the classes. Here, can you see? There are 3 types of classes: local, Edexcel, and Cambridge. Go to local. We are local syllabus. Once you go to the local syllabus, here you can see the grades. You all are grade 11, 2026 O-level batch. We have separately paper classes. I'll talk about the paper classes. And now we have grade 11 science, maths, commerce, ICT, and Sinhala. So you can purchase any of those class. Science is done by me. I am Dr. Rihan Rahim. Maths is done by Rizlan Hassan sir. He is known as Master Maths in Colombo. He is the teacher who has produced the highest number of A's in maths for the last almost last 15 years. He is the highest results producer in the Colombo area. Okay. Then commerce. Okay. Faizan Jifri sir, he is one of the business analyst. He is only doing the commerce subject. He is doing A-level business studies also, and one of the superb teacher. And ICT, Yusuf sir, Yusuf Riyaz sir. He is only, now see, our website was made by this sir. You can see how well this website is. Here you can, how to purchase and all those things are there. And now I can't purchase because I am not signed in. I'll just show you the classes. Then Sinhala is done by Ramesh sir. Then we have another Sinhala teacher, Madhubhashini miss. And English literature is done by Mazahira miss. I think she's still, she has not added the classes or anything. So Ramesh sir, Madhubhashini miss, they are also really good. But Yusuf sir, Islam sir, Faizan sir, I know them very well. They are my close friends also. So they teach the subject in a superb, proper way. They give the tudes like me only. They are giving tudes. They are giving the, they have the online classes. They have the recording. And we don't do hybrid classes. That means while we are doing the physical classes, we are showing it to the online students. In those classes, we are giving more focus to physical students. Online students are not cared. They are neglected. That's why we have started separately online classes, our full attention for the online students. So you can purchase the grade 11 classes when you go to this. Now, yeah, once you sign in, now I just randomly went. Now, once you sign in and all, it will show you the proper thing how to purchase. Now, my password is wrong. I don't know. Last year for the students, I made just a dummy thing and I just showed it to them. Okay, now you all know now if you all go to this. I don't know why it's getting loaded today. You will be able to purchase. So it will show first week, second week, third week, fourth week. January month, you can purchase the third and fourth week only because first and second week is free. No, recording also I will give. So you can purchase the third week and fourth week. That will be half fees. So 2000 rupee, you can pay 1000 and you can purchase the third week and fourth week. And you all can join the classes from the website itself. Paper classes, This month it is totally free, so I'll be sharing the link in the group, so don't need to worry. Next, same like this, you can purchase the paper classes. My paper class is there, Rizlan sir's paper class is there. Okay, next time when I'm showing you, I will show you now. Now this my password is wrong because it's an old account. I just made an account to show the students. I can't remember that password. I have to ask Yusuf sir and only get it done. So basic idea you all know. No? So you all can purchase. While purchasing, if you have any doubts, you can contact me. Okay, let's go to sclerenchyma, our last simple permanent tissue. Can you give what is this? Examples of plants with collenchyma tissue based on their function? All the herbaceous plants, all the dicot plants, they are having the collenchyma. If you take any leaf, dicot leaf, the veins are made out of collenchyma. They are helping in photosynthesis. They are giving little bit of strength to the leaf. And in the herbaceous plant, the young stage of the stem, they are having collenchyma. One is they are giving support, other one is they are helping in photosynthesis. Then sclerenchyma tissue, this is composed of dead cells. Now this dead cell is very important. I have to explain a very important concept related to it, so listen to it carefully. Cells are dead due to the secondary cell wall containing lignin. Lignin present in the cellulose of cell wall, not the primary. We call it secondary. This is the symbol for secondary cell wall. They have something called a chemical called lignin that makes the cell dead. As the cell is dead, there is no cytoplasm, no organelles, nothing. It's just empty. That empty space is known as empty lumen. There are two types of sclerenchyma. One is fiber type, other is chloride type. I will show both. Yeah, so look at this fiber type. Okay, if you read in the text, in the theory, in the tute I have given 5 polygonal in transverse section. So they look like this, polygonal. Okay, I'll tell you the story. It was primary cell wall. And all the other parts were there and it was living at one stage. This is the primary cell wall. Primary cell wall. Now itself, look at the diagram because after drawing, it will be like, different. Primary cell wall. Then after some time, what happened when the plant is growing, secondary cell wall grows inner to the primary cell wall. Secondary cell wall grows inner to the primary cell wall. This secondary cell wall had a special chemical. What is that name of that chemical? Lignin. Monada? Lignin. So what did this lignin do? Secondary cell wall it has lignin, cellulose and lignin. This lignin will prevent the water going into the cell. Therefore, all the internal parts will die and the cell, middle part of the cell will become empty. After it become empty, we call it what? Empty lumen. Are you clear? So finally, when you're seeing a sclerenchyma fiber type cell, you will be able to see like this. Primary cell wall, secondary cell wall, and the middle it is empty. Okay, this is the structure of what type? Fiber type. Fiber type of sclerenchyma. And sideways when you're seeing, it's like this. Fiber. This is the longitudinal section. From the middle we have cut and we have seen the inner part. That is only I have given up. Transverse section. This is longitudinal section. Very simply, in the coconut, you have this fiber, no? Coconut husk. Those fibers are what? Sclerenchyma. And we call it coir. We make coir wires, coir carpet. In Tamil, we say tumbu. That tumbu is one fiber, sclerenchyma fiber. Clear? Now let's go to the next type, scleroid type. In the scleroid type, it is circular cells. Primary cell wall is there. Initially, primary cell wall is there. And all the other living things, the vacuole and all those things are here. Then after some time, secondary cell wall will start to grow. Once the secondary cell wall start to go, what is there inside the secondary cell wall? What chemical? Lignin. Secondary cell wall contains lignin. Due to that, all the inner parts will die and become a empty lumen. After all the things die, we call it empty lumen. I'll draw another one. So after everything is done, we will be able to see a hole like this. All these are the primary cell wall and the secondary cell wall. Nothing inside. Empty. Because of lignin, the inner part of the cell dies. You might think, "Ayo, cell die, no use." No, for the plant, they need some dead cells to do a lot of functions. Our next topic, xylem, phloem, you will understand how do these dead cells help the plant. Okay, dead cells give a lot of support to the plant. They transport many things. All that happens. Don't think if the cell is dead, it is useless. No, in plants, even dead cells are useful. Therefore, these look like stones. They are called as scleroid cells or scleroid tissue coming under sclerenchyma. Nickname for them is stone cell because some of them are circular, some of them are like this, they look like stars because they have something called the pits like this, small canals like thing. So from the microscope, when you see these canals, it looks like stars, no? These people also have canals. These are known as pits. Small canal. Okay, you all don't need to much know about this. I'm just telling you. When you look from the microscope, you will be able to see these canals also. Because when looking from the microscope, they will put a dye, no? That dye will fill these canals. So nicely you can see. So some scientists, they thought they look like a star. They put star shape. But what is the actual shape? Circle, no? Spherical shape. That's why in our syllabus, they call them either spherical shape or star shaped. Can you all draw these two? I have kept two spaces in page number 9 to draw these two things. The fiber type of sclerenchyma and scleroid type of sclerenchyma. If you have any doubts, please send a chat box message or you can unmute and ask. For now, for today, we'll just keep the chat box on. From next week onwards, only the paid students will come now. So I will give the unmuting option. You all can unmute and ask doubts also, no problem. Yeah, better to take a screenshot. Later, you all can draw it. This has to come in page number 9. Page number 9. I have kept two topics. Fiber one side, scleroid one type. You can draw the diagrams under that. Scleroid, the circular cells. and then the secondary cell wall will grow and kill all the middle things. It will become empty. So this whole thing is the primary cell wall and the secondary cell wall together. In them you have small canals like this. Those canals are known as pits. So from microscope, when you're seeing, it looks like a star shape or spherical shape. Fiber is already polygonal, no? Primary cell wall and the secondary cell wall grow. Secondary cell wall will kill all the internal things and the middle space will become empty. We call it empty lumen. I hope you all are clear about it. If about this, if you have any doubts or any problems, just ask me. So what is the special thing present in the secondary cell wall? Lignin, very common question. Term test, surely they will ask. Why is the cells dead? Sclerenchyma cells dead? Because the secondary cell wall contains lignin. Because the secondary cell wall contains lignin. Are you all done with this? Yes, what are the functions of this sclerenchyma? Because they are dead, they are very strong. Because they are dead, they are very strong. So what will lignin do to them? Is it will kill or what? Actually, lignin will block the water going into the cell. Then what will happen? All the inner parts will become dead. Inner parts will die and become like an empty tube. So it will become like a pen. Okay, inside without the tube, if the pen is there, outer cover is there, inside empty space, no? That empty lumen. Okay, like that. It will become empty inside. Here, can you see? These are a diagram from the internet. The empty lumen, and these are the pits. You all don't need to worry much about the pits in our syllabus. This is fiber type. This is, they have taken a longitudinal section. This is the sclerite type. See, in the middle, it's empty. Yes, locations. Xylem and phloem, they contain fiber. Next, our topic is xylem. No, we will study in that. Xylem fibers are there. Phloem fibers are there. So, locations of fiber, xylem and phloem. Another location of fiber, these two are related to fiber type. These are related to sclerite type. Coconut fiber, agave fiber, cotton wool. Even in the mango, once you eat the mango, you have fiber things, no, in the seed. All those are falling under fiber type. All these are given in the tute, page number 9. These two are given under the fiber example. These three are given under the scleride example. Endocarp of the coconut, camphor, anger, and the mango fruit. Again, around the seed, that hard coating is there. That hard coating is made out of scleride. On the seed, you have this brush-like thing. If you take a mango seed, like this brush is there. That is fiber. Wait, I'll show the pictures, then you will understand it better. Pericarp of guava and pear fruit. When you're eating the guava, we say pera, and the pear's fruit, when biting it, you get a small sand-like feeling, gritty feeling. That is due to scleride type of parenchyma. And also in the coffee seeds and date seeds, coffee seed coating and date seed, ichapalam, okay, that seeds coating is scleride. So if you remember few of these examples, that is okay. I'll show you the picture. Now tell me, is it scleride example or fiber example? Fiber, no? Fiber example. The coconut fiber, from that only they make the coir wire, the rope, coir carpets and all those things. Bottom one is the agave fiber. They are like aloe vera plants, but not aloe vera plants. Okay. From them, from that also we can take fiber. In African country, they make fiber from those plants. In Sri Lanka and all, coconut is available now. So we make the coconut fiber. They are making the agave fiber. Here you have the scleride on the shell. And here we can see both. Inside that seed part, we have the scleride, and this fiber part, it's fiber. Okay. Nool wagere like thread parts. Agave is a plant. From that plant, this is the agave plant. From that agave plant, we can get agave fiber. The pericarp of the guava, that means the guava plant's outer coating, we can say, we peel it off. And even in the pears plant, you can see some dots like this, no? That's due to scleride. Sir, inner flesh of guava is pink or white? There is white guava also, pink guava is also there, no? Nowadays, it's mostly white, but pink is like the original one. Those are small guava. The big guava it's white. Kilo pera we say, that is white, no? Pink one is small and very tasty. Here you can see date seeds and coffee seeds. Actually, this is the coffee fruit. After that, you remove the seeds, you dry it, you roast it, you further roast it and make it coffee. Okay, here we are not talking about how to make coffee. I'm just showing you, even from the date seed, we can make coffee, no? That means this scleride part, I think, that's useful to make the coffee. I hope you all are clear about it. So last 2 points, we'll go to the functions. Yeah, it's almost 7. There are 2 functions. Mechanical support. It provides mechanical support and strength to the plant. It provides mechanical support and the strength to the plant. And also because it is very strong, because when it is dead, it is strong. It protects. So you can write protects the internal structure of plants. Protects the internal structure. For example, the coconut shell is protecting the soft inner part, no? The coconut shell is very strong, but inner part is very soft. So it's protecting, that shell is protecting that inner part. Like that, sclerenchyma can protect the inner parts of this plant. So Buddha, with that we are done with parenchyma, collenchyma, sclerenchyma. Next week, I hope to finish the xylem phloem and go towards, okay, now little bit we have to increase the speed because in our physical classes, last week I completed this your parallel class, we completed xylem and phloem. Because online, the link problem and that and this happening, no, from next week, no problem, no free link, no. From next week, you all can purchase the classes. Then immediately once you come to the classes, by 5, within 10 minutes, we will start. Full discussion, full explanation, okay? And make sure you have your pen, pencils, tutes, everything ready. And because next week we have to draw xylem, phloem, lot of drawings are there. And make sure you draw it very fast. If you want, you can have a rough book. First, you can draw it in the rough book, and later you can draw it in the tute. If you want, you can do it. Okay, then again, when you're drawing in the tute, you will get a double understanding, no? So you can do something like that because some students, they are scared to directly draw in the tute and, erase, and that they think that the tute will become untidy. If you feel like that, you just draw in a rough paper and later you can draw those drawing into the tute. Relaxedly, you can color and do everything because this tute is more organized. Okay, it will help you during your term test because now see, my next page. Here I have given a space. Don't know whether it's clear. Okay, you will be able to write parenchyma, collenchyma, sclerenchyma, all the features like a short note. Okay, so like that, it's well organized. Okay, it will be more helpful when studying for your term test because textbook you open and see everything is jumbled here and there, too many letters. Okay, mine is very organized, so maintain the tutes properly. If your friends ask you to teach, now you understood a lot of things, teach them, help them, promote them to our classes. Okay, if they want to join, tell them go through the website and join. They will also get the tute. Okay, so make sure you all do the needful and keep on studying. So there is no first term. First term is cancelled. Why is that? Already third term cancelled. Because of that, you all didn't study the grade 10 third term. I don't know why this is happening. Don't worry, I will have first term exam. I want you all to do the first term exam. Who is calling? Yes, any other doubts? My God, why is that? If they have the exams only, you all are studying, no? International schools, they are having it. But I don't know, government schools, there is an issue going on. Okay, anyways, no problem. We will start our paper classes also, no? Already we have started. Paper classes will happen tomorrow, 8 p.m. 8 p.m. we have our paper classes. So you all can join. It's a free class. I mean, for the whole month, this month we are giving freely because next month during Ramazan, we might not be able to have the paper classes because the prayer time, everything is clashing. So again in March, we will be starting. Okay, so at that time you all can purchase, but this month you all can come freely to our paper classes. Okay, this month the paper class is totally free. So please be there in the group. Send me your name. The ones who are not there in the group, name, grade, school, and your area. Okay, so I will add you all to the WhatsApp group. And also the ones who fill the Google form, Most probably we will be sorting it out and we will be sending the tute. The ones who have not filled the Google form, you all also fill the Google form. The ones who have already filled, you don't need to fill again. If you can edit, add your WhatsApp contact number also, because in the post office, sometimes when your house is locked or when the address is wrong, they give a call and ask where your house is. Now that technology is there. So if possible, don't add again the whole new thing. For your old Google form, if possible, just add your contact number. I didn't give that question. Now I have put that question. You can add the WhatsApp number also. Then it will be better. Easily they will be able to deliver it. Okay, then students, prayer time is also there. So I will end the session. You all can pray and then inshallah tomorrow we will meet again in our paper classes. The ones who are not there in the WhatsApp, please send me a message. This is my WhatsApp number. Send me your name, grade, school, all those details. Okay, then the ones who are joining grade 10 classes, 7:30 we will start. After prayers, we will start the grade 10 classes.